In this video, I'm going to be talking about the CD27-cd70 axis and some of the foods that might be able to help to mitigate it. So basically, what it is that CD27 is a T cell receptor, and it engages with the CD70 on tumors, and then recruits TRAF 2/5 activating NF-κB. And NF-κB is not a good thing, especially if it's constant and ongoing. So the goal of the video is to reduce that. The problem with chronic NF-κB is that it will upregulate exhaustion markers. The main one here, my main focus is PD-1. If that goes on for too long, then the PD-1 on the immune cell will be more susceptible to binding to and interacting with the PD-L1 on the tumor, which is not a good thing, because acute NF-κB activation can be beneficial. It does promote T cell survival and proliferation, but as I said, a chronic NF-κB activation is harmful due to PD-1, PD-L1 axis eventually happening. Okay, the PD-1 on the immune cell is actually a programmed cell death protein, and it's expressed mainly on activated T cells and B cells and some myeloid cells, and it's a checkpoint receptor that inhibits immune activation to prevent overreaction. Then there's the PD-L1 expressed on tumor cells and also antigen-presenting cells, which are supposed to train the T cells. And the function of PD-L1 is to bind to PD-1 to send a suppressive signal to T cells. That's the issue with ongoing NF-κB from CD27-cd70 axis. The tumor will eventually find another way or another option to suppress the T cells via the PD-1, PD-L1 axis. Now, there are consequences from ongoing PD-1, PD-L1 axis connection, and less cytokine production, such as IL-2, which is not good because T cells need that. Also, less glucose metabolism, reduced T cell energy, because no energy, leading to T cell energy exhaustion. Chronic long-term effects, chromatin remodeling, so at the DNA level, suppressing AP1. AP1 is needed for IL-2 for T cells, but with the AP1 loss, the T cells don't have IL-2, so they're not going to be functional. Also, reduced cytotoxicity and anti-tumor activity. So the tumor intentionally with the PD-L1 causes the immune cells and T cells to lose their tumor attacking capabilities just with the PD-1, PD-L1 axis. Now, there's a food here that has an effect on the PD-1, PD-L1 axis, and that food is red cabbage. It has anthocyanins and also sulforaphane. Sulforaphane has been very well studied, and indole 3-carbinol also has a vitamin C and polyphenols. And red cabbage, with all of these benefits, is that it actually downregulates PD-L1 by reducing inflammatory signaling, because tumor PD-L1 is often driven by NF-κB, also STAT3. STAT3 is one of the main cancer drivers, and it can oppose STAT5 at the chromatin level, eventually shutting down STAT5, which is not good because STAT5 is needed to boost the immune system. So, red cabbage and its sulforaphane suppresses NF-κB, reducing pro-tumor cytokines such as IL-6 and TNF-alpha. The effect is inducible reduction of PD-L1 expression on tumor cells. Red cabbage also does oxidative stress control because high reactive oxygen species leads to tumor stress. That stress on the tumor leads to the upregulation of PD-L1. But anthocyanins in red cabbage lowers that reactive oxygen species. Therefore, the effect is reducing reactive oxygen-driven species or ROS-driven PD-L1 induction or upregulation. Also, by lowering intracellular ROS, it improves mitochondrial redox balance. That's critical for ATP, for the immune cells and T cells, because immune cells and T cells are a very high energy demanding cell. And that's especially important if they're being suppressed by NFKB. Also, red cabbage has a double whammy effect on immune cells and T cells, but in a good way, because since PD-1 is upregulated when T cells go through high oxidative stress, um, red cabbage supports antioxidant protection of T cells, therefore actually reducing the PD-1 expression on chronically stimulated T cells. Because they're stimulated by chronic antigen exposure, and they're supposed to be, but that leads to the PD-1 being expressed on the T cells as well. So red cabbage also supports mitochondrial function as well, which is good because T cells need ATP from mitochondria. And um, since the PD-1 is being reduced thanks to red cabbage, the T cells are less likely to undergo the, the suppression that comes from binding to the PD-L1 on the tumor, therefore reducing the chances of AP1 loss. Uh, and then the IL-2 being difficult to be revived because once the AP1 is lost and the T cells are exhausted, it's very difficult to reverse that process, and a lot of factors are needed, such as there can't be vitamin deficiencies, for example. So it's better to avoid that situation in the first place, and red cabbage is a great way to stop the point where if PD-1, PD-L1, and CD27 and CD7, if that goes on for too long, where it is hard to bring back the exhausted T cells. Okay, now what led to this whole PD-1, PD-L1 in the first place was at the start of the video, the CD27-cd70 axis. A good way to prevent that is with pineapple. Pineapple has a bromelain enzyme. Also, vitamin C as well may reduce inflammation. But the main part about pineapple is that it actually cleaves the surface proteins like CD27, therefore lowering the overstimulation. Because if CD27 is not on the surface of the immune cell, then it won't even be able to bind to the CD70 on the tumor cell, therefore avoiding the NFKB that led to the PD-1 PD-L1 axis in the first place. Because pineapple, it literally cleaves the the adhesion molecules or adhesion machinery that holds the CD27 surface expression or even the CD70 on the tumor, so that those parts are cleaved off and not showing. Therefore, there is no suppression between the tumor cell and the T cell. Okay, so a quick recap. CD27, CD70 axis leads to NFKB, causes PD-1, PD-L1 axis. Red cabbage has anthocyanins, sulforaphane, reduces PD-L1 expression on the T cell, also good for mitochondria from the red cabbage for the T cells. And then pineapple cleaves CD27 on the immune cell so that it is less likely to interact or to be suppressed by the CD70 on the tumor, reducing the NFKB. Also, if NFKB goes on too long, AP1 loss, therefore no IL-2, and leading to a very long, lengthy, difficult, grueling process of having to reverse the exhaustion caused by the CD27-cd70 axis and the PD1, PDL1 axis. So that's it for the video. Like, comment, share, subscribe, and I'll see you in the next video.